A researcher sits at a computer, preparing a sequence record for submission. Besides genomic information, the record contains a scientific name, a catalog number and a country, referencing its source specimen. Before submission, the researcher wants to find more information on this specimen, for example, to verify its taxonomic classification. So what needs to be done? Searching different information systems such as GBIF, which hold occurrences that might describe the searched object. The researcher types the 3 pieces of information into a search mask, but receives nothing. The researcher ponders and decides to remove some information from the search. The search results, though, overflow when some scientific names are omitted, overwhelming the researcher. Faced with the prospect of manually searching through other portals, the researcher braces for a time-consuming task. The question is, what if there was a tool to help find such specimens? Welcome to SpASe. SpASe is a web service consisting of both an API and a graphical user interface, which I will guide you through in the screencast. Technically, SpASe acts as a specimen search engine that browses biodiversity data aggregators and specimen information systems using user-provided input. Currently, it scours DiSSCo, GBIF, and iDigBio for records using varying combinations of a set of input parameters. But that's not all. SpASe evaluates the found records for their similarity to the input information, assigns a similarity score, and sorts the found records accordingly. SpASe supports several formats that are used in sequence submission workflows. Here is an example of an EMBL flat file that contains a number of sequence records. In this file section, you can find information about a specific source specimen. If a URL can be found based on this information, SpASe will insert it in here. Okay, let's upload this file and wait for the record SpASe will discover. In tables like this, SpASe neatly organizes your data alongside any matching record it was able to find. Each qualifier describing a feature of the source specimen is displayed as a column. For example, you can see that the metadata of all input sequence records include organism as a field which holds a scientific name. However, the first column, specimen URL, lists resolvable specimen links discovered by SpASe based on the content in the other columns. By clicking on the arrow on the left, you can open the search result for a particular row containing records found online. In the specimen URL cells, the colored dots are an indicator of the maximum similarity score in the search results. A green dot stands for a score value 1, meaning that at least one perfect match was found. Instead, a yellow dot means the opposite. Right next to the status dot, you may also find a link icon showing that at least one URL is present in the result. The suggested records sorted by their similarity score can also be highlighted. Rows with links added to the parent row are colored green, while orange as row color indicates that the suggestions do not contain a URL as occurrence ID, or a URL that is not allowed, for instance, because it is not yet persistent. Besides this ID, a scientific name, and a voucher, there is nothing provided in this view that makes the similarity score somehow understandable. To access such information, you need to click on the plus on the left-hand side, next to the data provider from which the record was obtained. This detail view includes additional record information about identifiers, the location, or the collection event in general. Also, you find a similarities list that sheds light on the calculated score. In this example, out of the 7 parts of input information, 6 are identified to be similar. While no similarity was found in the evaluation of the location text strings due to different spellings, the comparison of the other input values resulted in a fairly high score, indicating a strong chance of similarity. If you consider this record indeed to be the correct one, you can take actions to update the original input information. Thanks to SpASe, the URL of the top suggestion is automatically added to the input record. As an alternative, you can also remove this ID by clicking this yellow button. Also, if you prefer to add a different link to specimen URL, simply click the Add button in the second column. In addition to adding specimen URLs, SpASe provides a feature that allows you to add specimen vouchers from the suggested records to the parent entry. These vouchers are constructed by the web service, utilizing catalog numbers alongside institution and collection codes found in the proposed record. This feature facilitates the construction of vouchers with more triplet components than, for example, just a single catalog number. If you find the constructed voucher not acceptable, for example because it could not be validated, you also have the option to revert to the original ID. Since this voucher could be validated against the source annotation helper API, which was developed in the BiCIKL project as well, we keep this entry. After completing our updates, we can export the file in the same format as its input by clicking on the export button above the table. Once exported, we can review the file for the changes made. As you can see, the specimen URLs and modified vouchers are included, ready for submission to a sequence database like INA. Last but not least, it is worth mentioning the API access to SpASe. Serving as the core of the web service, this endpoint allows individuals and organizations to seamlessly integrate this service into their own systems, promoting interoperability. This Open API documentation outlines the supported search parameters aligning with the INSDC feature table specification and provides practical examples. In addition to the parameters we've already covered, you can also search using, for example, coordinates or altitude, identifying agents, and specify collection signal dates or date ranges. Why not give it a try?